Hello guys, I am Sagar Sonon and today I am going to discuss about the DNA replications. In this today's video, we can discuss about all information related to the DNA replication. Mean what is the DNA replication, structure of the DNA, function of the DNA, and all the information. So, in the molecular biology, DNA replication is a biological process producing two identical copies or the replicates of the DNA from original DNA molecule. Okay, so DNA replication occur in all living organism, acting as the most essential part of the biological inheritance. This is the essential cell division during the growth and repair of the cell or the damaged tissue, while it is also ensured that from the each of the new cell receive its own copy of the DNA, and the cell process that extends to property of the divisions. Okay, and from own copy of the DNA, the cell possesses the extended property of the division, which is the make replications of the DNA essential. DNA is the made up of double helix and the two complementary strands. The double helix describe the appearance of the double helix. Double helix describe the appearance of the double strand DNA, which is thus the composed of the two linear strands that run opposite of the each other and twist together to form. Okay, during the replication, those strands are separated. Each strand are the original DNA molecule, then serve as a template for the productions of the DNA, and its counterpart process referred to as a semi-conservative replication. As the result of the semi-conservative replication, the new helix will compose of an original DNA strand as well as the new desynthesized strand, and cellular proofreading and the error checking mechanisms ensure that the new perfect variability of the DNA replication. In the cell, DNA replication begins at the specific locations as the original of the replication in the genomes, which consist of the genetic material of an organism. Unwinding DNA at the origin of synthesis of the new strand and accommodated by the enzyme known as the helicase result in the replication frogs ongoing by directionally from the origin from the origin. A number of proteins are associated with the replications frog to help the to help initiation and constitutions of the DNA synthesis. Polymerase synthesis is a new trend by adding the nucleotides and that are complement each strand, and DNA replication occurring during the stage interface, and DNA replications also perform as a in vitro DNA polymerase isolated from the cells, and the artificial DNA primaries can be used to start the DNA synthesis as known as a sequence in a template DNA molecule, and the polymerase chain reaction, ligase chain reaction, and transcription mediated amplification are the example. Okay, so DNA exists as a double string structure with the both sides called coiled together to form the characteristics of double helix. Each single strand of the DNA chain of 4 types, that is nucleotide in a DNA contain the deoxyribose sugar and the phosphate and nucleobase, and the 4 types of nucleotides correspond to the nucleobase adenine, cytosine, guanine, and thymine, commonly abbreviated as A, C, G, and T. Okay, adenine and guanine are the purine bases, while the cytosine and thymine are the pyrimidine. Okay, those nucleotides form the 5' bond. Okay, so in the today's video we can discuss about the DNA replication as well as the DNA. So thank you.